gentlemen. It's a great pleasure to be here today and to exchange with you about personalized medicine in transplantation. So, what is about personalized medicine? We hear that term over and over. In every single grant application, you find the top keywords, you will find personalized medicine. And actually, that's true. We are doing personalized medicine in our clinical practice every day. And we are not the first ones, because personalized medicine had started years and years ago in ancient Greece, when the prevailing dogma was about to assign superstition to separate between health and disease. Hippocrates, of course, came with the rational thinking that actually the disease could be attributed not to superstition, but to natural process. Observation, examination, prediction, and adaptive treatment was the theory, the timeless theory of Hippocrates of personalized medicine. So, where are we today in personalized medicine in the 21st century? We move from personalized medicine to precision medicine. We have new tools, we have the genomics, omics technologies. There is not a slope increase, but an exponential increase of the knowledge and the biomarkers that we can be using of treatment, computers, powerful statistical models. And I would like to share with you in the next 20 seconds this important initiative that has been done and released in January 2015.
And that's why we're here today. Because something called precision medicine, in some cases people call it personalized medicine, gives us one of the greatest opportunities for new medical breakthroughs that we have ever seen. Doctors have always recognized that every patient is unique. And doctors have always tried to tailor their treatments as best they can to individuals. So the time's right to unleash a new wave of advances in this area, in precision medicine, just like we did with genetics 25 years ago. And the really good news...
The really good news is for cancer, unfortunately for us. Because when you read this New England Journal paper released in February 2015 in this new initiative on precision medicine, I read this paper carefully for preparing this talk, and cancer is cited 16 times. No need to say that end-stage renal disease, hypertension, or even transplantation are not even mentioned. But that's fair. We are not competing with cancer, but we can ask ourselves, is precision medicine adapted in the field of solid organ transplantation? Of course, the answer is yes. The prevalent organ transplant universe is about 1 million people worldwide having a transplant, with 120,000 new organs transplant per year and 50,000 organs transplant fail each year. So improving allograft survival is one of the major goals of transplant medicine. And we have to acknowledge and we have to face more complex phenotypes, more complex disease. We have pushed the boundaries for crossing the HLA barrier and have high-risk cohort. And it's not all about HLA. We have also expanded the spectrum of the kinase we are using, and we are using more and more, at least this is true in Europe, expanded criteria donor kinase. That's what we recently reported in the British Medical Journal. So, what are the milestones for precision medicine? Address transplant phenotypes across organs, reclassify disease states, engineer new diagnosis systems including new technologies, modelize individual risk prediction, and set the stage for response to therapy and clinical trials. And since we are not mouse doctor, at the end of the day we need to treat patients, so we need to apply that in patients, and we can use nowadays extensively phenotyped cohort of patients, transplant patients, prospectively enrolled in studies with a high level of detail, unselected, with timeline evaluation, histopathology, biopsy, sera collection that we can test at different time points. And a number of events are important to do some risk model prediction and individual risk profiling. So, if we get this population, and we are not inside registries, which can be useful, but we need to have precision diagnosis inside the cohort in which we run the model and the powerful statistical tools, like smart data, performance calibration, artificial neural network. I'm going to be talking about that later on. But of course, we are facing issues. The issue is that the current state of the art is facing misdiagnosis, incorrect classification of disease, no connection between specialties, and lack of data-driven approaches. So we need to move beyond the state of the art. We need to have integrative and smart data thinking of transplantation. We have to build networks, huge networks, precision diagnostic platform. We have to connect specialties and transplant specialties together. And we have a lot to learn about the other specialties. And we need to use those cutting-edge technologies as a prospective manner at repeated time points. We need multi-dimensional transplant assessments, as summarized here in this cartoon. So we are facing issues, but let's have another look on the new understanding and strategies that have been used for better assessing transplant outcome. The first step will be precision phenotypes. The second step will be precision immunology. Then we will see how molecular measurements in biopsies could inform the conventional features. And then I will cover some aspects of integration strategy and multidimensional equations for individual risk profiling. So we have to acknowledge the great contribution that the BANF classification has made in the field of solid organ transplantation. If we are here today in this room, all talking about the same language when we talk about kidney pathology and diagnosis, this is because of BANF. If you look at the BANF paper, among the 10 top-cited papers in HAT, you will find 4 BANF papers. And this is the last paper released in HAT in 2014, related to the meeting in Brazil held in 2013. And you will see the extension of the spectrum of ABMR, the inclusion of C40 negative ABMR, the recognition that subclinical ABMR and indolent phenotype do exist and do produce injury and late allograft failures. And last, the inclusion of antibody-mediated vascular rejection. This is a compelling evidence coming from many studies from Europe and the United States and all over the world, all converging towards these data that have been adopted in the classification. So, of course, we need also to decipher new phenotypes, and I would like to illustrate a recent paper that we published in Circulation Research Journal. If you don't want to read the paper, please read the editorial by John Bromberg, which really greatly summarized the strengths of this study and also its limitation, like any study. So, in this paper, we use transplantation as a human model of disease. This is why we published that in a basic science journal. And nowadays, if we have a good assessment of the cohorts and we use contemporary tools, we can isolate and we can not only report association, we can approach causality. Of course, it's not perfect, but it can be biological meaningful. So here is the study. The question was, is there a DSA-mediated component in accelerated arteriosclerosis post-transplant? So we evaluate this large cohort of 1,000 patients. We did an extensive workout on assessing donor cardiovascular profile, recipient cardiovascular profile, the drugs used, baseline transplant profile. And we had the chance to have this one-year protocol biopsy schema. And in the first year post-transplant, all this sera that we could have assessed for DSA and all these thousands of biopsies that were carefully reassessed for diagnosis to get precise diagnosis. We wanted to go beyond the traditional risk factor for accelerated arteriosclerosis, so you need to have an overview of what happens post-transplant in the first year in terms of lipid profile, blood pressure, CNI, etc. And you have a large input number of variables, but you can sort out that beyond the traditional factors, there is independency and a biological gradient for your antibody, depending on its strength or complement binding capacity, to be associated with accelerated arteriosclerosis. What about precision immunology? You've heard a lot about DSA, so it's going to be only one slide. But this is an important slide because the more we know about anti-HLA DSA, the more complicated we realize that the story is. DSA response is like chaos. The status of a single DSA can widely change with time, and now we are convinced that a multiplex assessment of DSA is mandatory. MFI levels, class, aplets, affinity, avidity, strength, preform, de novo, complement binding, IgG subclass. And let's not be depressed by the test. Every test has its strengths and limitations. But these tests were and are and will be very helpful to better connect the properties of anti-HLA antibodies to allograft injury. This is very important also to integrate that the timeline and the kinetics of a single DSA widely vary with time. And this is a mandatory thing that we need to integrate when we are interpreting studies. There is no doubt, if you take the literature and you do a systematical review or meta-analysis, and you include all the ongoing and already published studies across organs, you will find a complement binding capacity of antibody, whatever the technique is, are deleterious for the allograft. There is no doubt about that. And of course there are exceptions. It's not perfect test, and it has to be refined as well. But this is interesting to model the risk of graft failure and disease progression, risk prediction, and also it has to be validated prospectively. So I'm done with the essay, and now where precision medicine has to move. Are molecular measurements useful in the clinical setting? Are they able to inform biopsy and to correct the conventional features set by the classical histology? So in 2010, there was this array of hope for kidney transplant, and this editorial in 2013, the question is that, is it like more than a nice toy for researchers? What is the clinical relevance of all those biomarkers and approaches that you see in every issue of Transplant Journal? So we built this collaboration with the team of Phil Halloran in Edmonton, and the question really was, are integraft gene expression able to improve disease activity and stage inside the ABMR disease, in a very prototype and homogeneous cohort of type 1 early ABMR. Why do we use early ABMR? Because it's a prototype of disease when you know the onset of the disease, you know the risk, and it's a very early, so that you can get rid of all the interaction and the possible interaction and the the treatment management of patients. The hypothesis is that molecular graft measurements could enhance individual risk stratification in ABMR. So this is the study design. We screen patients for having early ABMR in 2 Parisian centers, in Necker and Saint-Louis Hospital, 128 patients included with early ABMR. I have to mention that they all have the same treatment, they all had the same standardized management, and also with protocol biopsy and etc. And we assess clinical, histological, immunohistochemistry parameters, DSA parameters, and we added these gene expression assessment in the biopsy at the time of ABMR. And we have follow-up biopsy so that we can model progression of TG and time to graft loss. So this is a summary of the data, and since you are using these variables, they are, of course, interaction, correlation. So you have to take care and be very careful of this correlation and interaction between variables. So what I'm presenting you is the set of genes that we are using in this study. We are not inventing gene set, we are using already published and validated gene set. A classifier derived by machine learning that you've heard of, which is the ABMR molecular score, and also the EDSA-ST, which is a metagene derived on previously published gene sets. So the story gets more complicated. We started with histology phenotype to immuno-histo-phenotype and now to immuno-histo-molecular phenotypes that are represented in this dendrogram. But you can sort out the question, and this is the main result of the study. When you model the risk prediction for allograft failure in patients with ABMR, adding the molecular microscope strategy and the ABMR and the EDSA-ST, you will see that having an old donor when you get ABMR is not good. It's a cumulative burden of vulnerabilities. If you have a bad GFR time of diagnosis, there's no surprise. If you have the intensity of your humoral lesions, including TG and microcirculation injury, are also independently associated with outcome. But look at here, the ABMR molecular score was also, at least in this selected population, an independent factor associated with increased risk of graft loss. We had to confirm the result in an external validation cohort, and we found consistency between these results. So now the question is it useful? In other words, when you add a molecular microscope in this set of patients with ABMR, what do you add in terms of conventional features for improving the individual risk prediction of failure? So this is NRI, IDI, so net reclassification improvement, etc. It's complicated calculation, but the principle is very easy. You take your cohort of patients, you split them in 2, patient without graft failure and patient with graft failure, and then you calculate for each individual the predicted probability of failure, and you look at the motion in the 2 groups. You're expecting that it goes in the blue, meaning that when you add the molecular microscope to the conventional features, you will decrease your risk assessment and your predicted probability of failure. In another hand, when you take the failing group, of course, you are expecting the red movement, meaning that you are increasing your individual prediction risk of failure. So you can summarize that and characterize, and you see that when you add a new tool to the conventional assessment, at least it could help you to reclassify the risk. So the take-home message is that if you take a cohort of well-phenotyped ABMR patients with similar histology, similar DSA, you can have different molecular signals, and the different molecular signals confer to different response of therapy and different outcomes. So we need to dig that in more detail, but this is a preliminary experience in ABMR, which is interesting. So last part of the talk, of course, integrative strategies. Now that we have all these variables together, we need to build that kind of network. We need to implement routine lab histopathology molecular phenotypes. This is smart data, and we need to run that in the iBox, which is the integrative box when you have all these inputs, and then you do machine learning, highly dimension analysis, and you can provide diagnosis, activity score, reporting, and response to therapy. And of course, you need to do some machine learning where the data and the reference set is enriched and implemented with new samples, with the mistakes that you can make in the new diagnosis or not. So, of course, we need to use fancy methodological tools because this has to be validated. As I said, the large number of input variables made that we are using artificial neural network with hidden layers that are predicting success or failure. We need to combine and have complementary approach, random forest, integrative nomograms. We need to do prediction, calibration, validation. It has to be bootstrapped, externally validated, and we do iterative loops to make sure that of the consistency of the association we find and the quantification of their amplitude, which is very important. So let's take an example. We'll move you through this particular patient that you can see here. So this is this guy, 1 year out of transplantation. He has a circulating DSA, which is the DR with a 6,000 and so MFI. He has IFTA2 and he has a degree of microcirculation injury, but it's very mild. G1, PTC1, C4D negative. Pretty good GFR, almost 60 milliliters per minute, and mild-grade proteinuria or low-grade proteinuria. Seems that it's pretty okay, but if you project and you run him in the iBox, the equation will see that your patient is in big trouble. He's 1 year out, but his individual predicted probability of failure in 3 years will be 69% survival, The allograft survival of 5 years will be 46 and 24% at 7 years. So what happens in this patient? He's stable, he has a kind of indolent ongoing process with a circulating DSA. He could have been called subclinical ABMR. So what does the molecular microscope say in this patient? And you can see here the summary of the reports that you can get and that we get. It's a moderately disturbed kidney with some features of AKI, but the ABMR molecular score and the rejection score are at the highest level. It's a full-blown ABMR with a lot of endothelial activation, endothelial injury, gamma interferon response, NK response. And when you project this sample in this PC analysis with the nearest neighbors, you will see that this patient is actually having an IBMR which is at the top injury of all that we can get and all that the reference set in Edmonton has a bad condition for this patient. Probably he needs a treatment. We don't have the answer yet, but still. So what about precision medicine? That's going to be the end of the talk. This is not science fiction. We are part of this project launched by Edmonton, the Intercom X ATAGC ongoing studies. And what we do at Necker and Saint-Louis Hospital, when you have a biopsy for cause, and it's going to be very useful for these highly sensitized patients that we transplant. So we do an extra core, and it's shipped at room temperature in RNA letter. And we get some molecular reports in 48 hours. We get answers, and we get some surprises sometimes. Sometimes there is agreements between the conventional features and the molecular microscope. Sometimes we see borderline lesions which we would like to treat with the steroid pulse and which obviously do not have any features of TCMR, but that's another story. So we need to be prepared for precision medicine in solid organ transplantation. Improving phenotypes and re-stratification, we are getting there. All societies and the impulsion in the BAMF classification have to think about implementing those new technologies in the clinical practice. At least try to consider them as a companion to the previous system that we are using. And for treatment strategies, maybe this approach, the iBox approach, the molecular microscope, could be also useful to include patients in clinical trials and to be a surrogate for treatment efficacy, for response to treatment. But there's some way to go, and I would like to thank all our partners, and Toulouse Hospital, Necker Saint-Louis Foch, Charité, and Adriana Zevi in Pittsburgh, and of course, Phil Harwan in Edmonton. Thank you for your attention.